Hello guys, so in this lecture we are going to talk about DNA replication and repair. So as we know, we've been talking about cancer biology and what are some hallmarks of cancer. And one of the big hallmarks of cancer is genome instability, which starts partly because of mutations that happen initially to start this process, to jumpstart this process, that are in one of the key pathways that are somehow related to cell cycle control, growth control, or DNA repair. So these are some of the main pathways that are known to be mutated in cancers. And so it's very important to understand the way DNA repair works in a normal system before we can step back and look at how it is altered in a cancer system. So before we do that, let's go ahead and review replication machinery inside our eukaryotic cells. So as you know, the DNA replication doesn't happen in a vacuum, and it doesn't happen with just a single molecule in place, but rather it involves a host of different proteins and molecules coming together in order to help us replicate the DNA appropriately. So you have your helicase that's coming in and unwinding the DNA. You have the single strand binding proteins keeping them apart, keeping the two strands of the DNA apart. Then you have your topoisomerase that's relieving the tension by actually creating nicks in the DNA itself so that it can relieve the tension, so that there are no random breaks in the DNA as a result of the over bending of that structure. Then you have the DNA polymerase that is obviously replicating the actual DNA, but it also has that proofreading capability. If you remember, in that horseshoe shape, you have the catalytic site for replication and then a catalytic site for proofreading. So you have these slider clamps, you have the RNA primases, you have a host of different molecules coming together. And so there's a lot of players involved, right? There are a lot of players involved. And when this machinery is in full form working to replicate this entire enormous amount of material, right? It's billions of base pairs of material that they are replicating. There are bound to be mistakes. So there has to be a system in place to fix those mistakes when they happen, primarily because there are dire consequences to leaving those mistakes in place. A single nucleotide change sometimes is enough to cause a very, very harmful phenotype. As you can see in sickle cell anemia, it's just a single nucleotide shift in the beta globin gene, and that's enough to change the shape of the red blood cells so that they don't flow as well. They stick to each other and aggregate, leading to a higher rate of clots. They don't bind hemoglobin well, so they can't transport oxygen well enough. So there are lots of possibilities that can happen, very bad things that can happen if you don't fix those mistakes. Now also, you can, if you don't repair, if you continue to leave the mistakes where they are, over time they can have even more severe consequences for a cell or organism in the form of cancer or another phenotype where the organ system is unable to work anymore because of all these mistakes that have accumulated. Now, there are two main types of DNA damage that can occur inside our cells. One is what we would call an endogenous damage, and this occurs in the cells without any outside influence and is usually a result of either replication error as the DNAs are getting replicated, or it could be due to spontaneous changes in base structure, which happen all the time, by the way. So that's something that's always happening and always fixing. But it's one of those things that the base can change, or base can bind to each other, cause dimers and things like that. They can get cross-linked, different, the different strands of DNA. So lots of things can happen like that. But there has to be a system in place to fix it as well. And then strand breaks, obviously. Now, these strand breaks primarily happen at replication fork spontaneously, again, because as the helicase is unwinding one side, another side is getting overwound. But it could be through other mechanisms as well. It could be through oxidative, reactive oxygen species just running around and causing these breaks as well. Now, exogenous damage, on the other hand, are damage to DNA that is induced by an external or environmental source. And so these would include all those chemical mutagens or chemical carcinogens that we talked about in cancer biology introduction that can alter either the structure of DNA bases or bind to the DNA in some way as to hinder it from remaining the way it should be. It could also be a result of radiation or chemicals that can directly cause either dimers between adjacent pyrimidines or actually cause breakage of DNA strands. This could be a single strand break or a double strand break. As you can see with UV and ionizing radiation, we'll find out a little more about it in a second. So one of the things that we need to be mindful of is, well, what is exactly that's causing the damage that we're trying to fix? And then we can look at what type of repair pathway can fix that damage based on what has happened as a response to these, different factors that can contribute to DNA damage happening. Now, the DNA damage, as I talked about a second ago, can occur all the time. It happens all the time, even when there's no replication happening, even when there are no mutagens available, just living their life, just cells doing what cells do. There will be constant damage occurring in the DNA, but it's constantly getting fixed as well. Now, two main types of damages that kind of happen spontaneously all the time are depurination and deamination. Now, depurination is specifically when one of your purines, one of these double ring bases, get taken off the DNA strand. So it just gets removed. Just again, they are in an aqueous environment. The water, oxygen in the water can react with oxygen in the actual amino acid, in the actual bases, and it can cause it to get removed, right? So you end up with sugar phosphate inside your sugar phosphate backbone. You end up with this hydroxyl on one side instead of its normal where the base would be. And so that can cause your base to be removed. Another thing that can happen is deamination, where a cytosine can have its amine group removed, this ammonia removed, again with the input of water, leading to the oxygen getting replaced there. That changes it from a cytosine to uracil. Now, obviously, if this was right at that time of replication, and this happened to be the case, then instead of a C, when the DNA is replicated, it would assume that it was indeed a T and place an A across from it, which would change the fidelity of that strand, that new strand that's forming. Another type of, and this is now, the first one was just looking at endogenous, just normal DNA damage that's happening all the time. Now, another type of damage that's, again, going to be happening all the time because all of us have exposure to sunlight and the UV radiation that is in there, and that is the formation of what we call thymine dimers, where two adjacent thymines can bind together and form these dimer structures based on their interaction with each other. And this cross-linking happens due to UV radiation. And this, again, is going to be where causing these dimers is going to change that DNA structure in a way that it can no longer be replicated properly. So these chemical modifications of nucleotide, if obviously we leave them the way they are going to lead to mutations. And that's obvious just looking at it. But here is, if you look at it a little bit more closely, the case of deamination, if you are looking at a C to U conversion, while a U or a uracil is not going to be able to bind to a G the same way, it's not going to be able to have the same hydrogen pairing as before, and that's going to cause the DNA to actually, kind of bulge out and be not in its appropriate conformation, which is actually how it's recognized as not being appropriately placed. However, if this was replicated before it was fixed, one of the strands is going to be unchanged, so it will give rise to a completely normal replicated sequence. However, On the opposite side, where you have the U, it is going to now, the new strand will contain an A instead of a G. And obviously this would lead to then a mutated sequence. Now this mutated sequence only affects that particular base pair, right? Just that one single nucleotide change. So while if it was in one of the coding regions of the genome, while that may affect, not that it would necessarily, but while that may affect the actual codon, that particular amino acid, everything else downstream of it and upstream of it should remain unchanged. On the other hand, you have a situation where it's depurinated, or where you have a removal of A because of depurination. In this case, one of the new strands again remains unchanged and has everything the same, but the second strand where the A was removed due to depurination, now when it's replicated is going to be missing that base pair entirely. And in this case, everything downstream of it will be affected and will be disturbed and will not work appropriately. So that is a case where if it was in the middle of the coding sequence, all the rest of the codons downstream of that particular mutation are going to be affected and probably leave that protein useless. Okay, so that's one thing that we need to remember, how deamination will affect the genome versus how depurination will affect the genome, right? Those are two very different things, and the way the results can be very different as well. Now, obviously that means that any errors that are made, whether they are made during replication or outside of it, must be corrected, because you don't want to mutate important portions of that genome, right? So that is why the DNA polymerase has that ability to proofread as it goes, and kind of that's an internal balance inside it, right? In addition to that, you have other ways to correct it as well, but at least at minimum there is that internal check and balance system within the replication itself that tells you how important that process is. Here you can see that if it wasn't the case, if you didn't repair mistakes that had happened in the parent DNA molecule, then as the DNA is replicated, the original from the original parent, you're going to end up with a permanent mutation in one of the cells, because at that point, once that strand with error has been replicated, that's the new strand, that's the new sequence, that's what's going to get replicated every single time. So that mutation is there to stay then, and that's extremely important to understand, that it is you have to fix these mistakes in a timely manner before the cell has a chance to replicate, because once it replicates that mistake, that is there to stay. It's a permanent mutation inside that new cell. And we can see that how accurate DNA replication and repair process is by comparing genomes from across different species of the same gene, same gene sequence from different species, different organisms, and also just over time in humans or another model system. And you can actually record the fidelity of the DNA replication through that by looking and seeing that a particular gene that is conserved in all the species has majority of that section conserved, right? The very few changes that are made in many of the genes, and those changes can generally be attributed to one of those important processes that we just talked about, the deamination and depurinations, because you can see the shift that has happened, right? The T to C and A versus G, G versus A. That would be typical of those type of mutations happening and getting lost. Now, how does a cell fix the DNA damage? Well, DNA damage can be fixed through multiple ways, and it depends on how DNA damage had occurred and what type of DNA damage had occurred before we can see what type of repair mechanism is going to be used. So if you have a DNA replication error, or an error due to stress that's just a single base pair mismatch, or a single insertion or deletion, something just involving a single base pair as the resulting damage, then usually the DNA repair mechanism that is taking care of these is what we call a mismatch repair, because that's what it is. It's a mismatch when there are two base pairs against each other that shouldn't be against each other. In another type, if you look at when you have a lot of reactive oxygen species, or you have exposure to ionizing radiation or chemotherapies, that can lead to single-strand DNA breaks. In that case, you usually use base-excision repair, where you need to now, again, take care of that base that has been damaged or broken up. On the other hand, if you have exposure to chemicals that are polyaromatic hydrocarbons or UV light, that may lead to some kind of DNA adducts adding to the DNA strand, intercalating inside it, or causing a mutation through that mechanism, causing some type of cross-linking within the strands, then there needs to be a little bit more excessive repair. More region has to be removed, and that's when we when our cells use what we call nucleotide excision repair. So base excision repair versus nucleotide excision repair is what we'll talk about soon afterwards. And then that's usually the mechanism that we use when we are looking at transcription coupled global genome repair. So anytime there's transcription happening of those key proteins, that's the type of repair that's usually taking place along with it. And then finally, you can have what we call double strand breaks because of exposure to chemotherapeutics or ionizing radiation, where both strands of DNA have broken up. And in that case, there are two different ways that they can be fixed. They can either be fixed through homologous recombination, or they can be fixed through non-homologous end joining. Okay, so now we're going to go through and kind of talk about the basics of each one and when that would be used, and how exactly it works. So the first thing we'll talk about is base mechanism of DNA repair, right? So just the basic mechanism. How does it happen? No matter which method we use, the three main steps remain the same. The first is excision of the damaged base or damaged area of the DNA strand. And then the second step is obviously to repair that damage by replicating that portion of the DNA. And then finally, you have to always seal the nicks and ligate the backbone so that it is a smooth strand and does not contain any knots or breaks. So looking at the first one, the simplest one, which is DNA mismatch repair. In DNA mismatch repair system, you remove the replication error that has escaped proofreading. So this is usually an aftermath of DNA replication. So DNA replication, whatever got missed by the DNA polymerase's own proofreading mechanism, will get taken care of by the DNA mismatch repair. In this case, when it notices that there is a mismatch in the replicated DNA, leading to obviously that change in the shape of the DNA by, because of non-appropriate hydrogen bonding, it will then remove that portion of that base pair, and it will read and refix that original sequence to restore it. Now in this case, remember, it always removes, it could have removed either one, but it always removes the newly synthesized strand. So that makes sure that you are taking care of that error, because that's where it's more likely to have occurred. Now in mismatch repair, the way it does is that it's not just removing that single base, but rather it's going to remove a small portion of that newly synthesized DNA. With the help of what we called DNA mismatch repair proteins. They work together where one of them binds to the DNA mismatch, the place that is not in the appropriate conformation, while the second one makes a nick in the further downstream of the DNA on the new strand and binds to that. and that whole section is then looped out and removed from the replicated DNA. Now, again, it's only removing the newly synthesized strand and not the parent strand. And then that repair of the gap can be easily repaired just like usual with the DNA polymerase and ligase. Now, in this case, no primase is needed to create a primer because you already have overhangs on both sides that can be used to fill in that gap with the DNA itself. Now, when there are other mistakes that are happening, like we talked about, that things happen all the time to damage the DNA, there are two main excision repair pathways that fix single strand breaks. So just single strand mistakes, rather, not breaks, but mistakes. This is looking at either deamination damage or pyrimidine dimers of some sort. When this is a deaminated damage, the process that we use is called base-excision repair. In this case, the first step is only to remove the base that was damaged. Nothing else, just that single base that is damaged, that is not supposed to be there. For example, uracil will be removed from the DNA by DNA glycosylase. Once that is removed, remember the backbone is still intact. Another enzyme called AP endonuclease would come in, and then that, with the help of phosphodiesterase, will remove the sugar phosphate backbone again just from that single base pair. And then the DNA polymerase will add the new nucleotide, and DNA ligase will seal the next. So it's just correcting a single base pair that has gone wrong, not worrying about anything else around it. On the other hand, in a nucleotide excision repair, you remove a more broader piece of that DNA strand that is damaged. Again, in this case, there'll be two pyrimidines that are right next to each other that have cross-linked and bonded to each other in a way that is now not going to allow them to separate and work appropriately. However, in this case, the nuclease doesn't remove just the dimers, but rather removes an entire segment of up to 29 nucleotides, where it includes the bases as well as the actual backbone. So the entire section is removed, and then DNA helicase will again keep that area open, unwound, and the DNA polymerase will come in and correct that lesion and fix the problem. So this is a way to correct what we would call the bulky lesions, right? Because they've cross-linked, so they've caused like kind of a kink or bulky portion in the DNA that would have distorted the DNA structure, right? And so that's how we would have fixed those type of damages. Now, similarly, there are two different ways to fix double-strand breaks in the DNA. Again, those can be caused by certain chemotherapeutic or mutagenic chemicals, as well as by ionizing radiation. In this case, there are two different ways that they can be used. One way, and these could, by the way, also be accidental double-strand break. It doesn't have to be because of some other mechanism. In an in a non-homologous end joining, specific nucleases will chew up some of that region so that it is opened up properly, and then it is repaired, so some of that region is just removed and never filled in, essentially. And then ligase will come in and join those ends back together. The problem is this is going to result in loss of nucleotides. So this is not going to keep the sequence the same, right? It's destroying some nucleotides permanently and then joining the ends back together again. On the other hand, you can have what we call homologous recombination. In this case, you need the homologous chromosome with its intact DNA sequence in place. And what you get is processing of the broken ends by what we call recombination specific nucleases, which make sticky ends, what you can call sticky ends, right? So there's a single strand. So it only chewed up partially one of the strand. And then it can be accurately repaired using the undamaged strand as the template. And then you get a completely filled up, appropriately made DNA molecule with no loss of nucleotides. So non-homologous end joining is not accurate because it leads to loss of nucleotides and possibly mutations that can have lifelong implications, while homologous recombination is accurate. That's the way that you would want double strand breaks to be fixed. So in homologous recombination, you flawlessly repair DNA double strands, because what the nuclease does is that it only chews up one side. It chews up the 5' ends of the broken DNA strands on both of the molecules, so that it creates what we call sticky ends. Now these sticky ends can then, it's a little bit like crossing over type structure, where it then combines with the homologous chromosome, and a repair polymerase, a different kind of polymerase, repair polymerase, will synthesize that DNA in that using the undamaged DNA as their sequence, as the complementary DNA sequence. And then once it has passed through that mistake portion, the invading strand will be released, and just simple complementary base pairing will then fill up the rest of the break. So one of the strands is getting filled up using the complementary strand, and then using that filled strand as the template, the remaining strand is going to get fixed. And then you again have your DNA ligation happen just like before, and thus repair that strand accurately. Now, DNA repair pathways are commonly, many of these pathways are controlled by tumor suppressive p53, which we talked about in the last lecture, that is one of the most mutated pathways in cancer. Almost all cancer shows some mutation or some reduction in its function at some point in their, structures in their genome. So p53 is what we call a master regulator. It controls responses of our cells to various types of environmental cues, including but not limited to DNA damage, hypoxia, starvation, exposure to heat or cold shock, genotoxic drugs, UV, gamma radiation, any one of these things. It's a cell cycle control protein, which we will learn about how it works at the cell cycle checkpoint. And it also can induce DNA repair pathways when there is damage to the DNA. So p53 can help with a lot of different types of repairs related to DNA damage. When it is unable to fix the damage, what it will do instead is induce apoptosis in the cells, or program cell death in the cells, to then let you know that, this cell is no good anymore. So those are some of the pathways, again, that are going to be controlled by p53. Now, you don't need to know, obviously, all these key players inside every single one, but you do need to know that p53 is a master regulator and can regulate and affect multiple parts of cell regulation as they pertain to cell cycle and DNA repair and apoptosis. So one of the things, and I'll show you a couple of examples of that, right? So in the first example, we're going to look at how p53 will respond to single strand damage. If the damage is mild and it can be fixed, then p53 interacts with downstream regulators, mediators, those second messenger molecules like MDM2 and CBP, and cause a growth arrest response, while also activating DNA damage repair pathways so that they can fix the damage and the cell can still recover. However, when this damage is extremely severe, different proteins are activated before p53 is activated itself, and these alternate mediators essentially signal p53 to induce an apoptotic response instead of a growth arrest response. So while in both cases MDM2 is released from P53 to activate it, P53 itself activates different proteins depending on what it is that it was, activated by and what it is that it mediates or modifies itself. So again, reminding you, in a signaling cascade, you have an incoming signal that is going to go through mediators that will modify other signaling molecules in the cascade that will eventually enhance the signal to have the final impact in the form of effector proteins leading to the response. So the effector proteins are what cause the response. And all these in-between steps are your mediators of the pathway. Now here another example is where you are getting response to double-strand break by, again, the same exact protein P53. In this case, ATM, another signaling cascade, right, another mediator, another protein that is first going to be activated, is going to activate P53 either through check 2 or by itself. And depending on how it is activated, it is going to then either cause growth arrest and DNA repair to get activated, or apoptosis or senescence to be activated. Again, the incoming signal would be whatever damaging agent has come into contact with the cell. The mediators would be these proteins that are activated first and that are then going to regulate the core regulators, which is P53, that will then regulate its effector proteins, leading to the response that we see at the end of the day. So the importance of DNA repair pathways is, clearly seen when we look at how as we age, DNA mistakes obviously accumulate. So you have a higher chance of incidence of cancer. And also when we look at P53 protein directly, it is directly mutated in over 50% of all human cancers. And when you combine the indirect and direct combination, you see that almost all cancers show some type of mutation in the DNA pathway. So just to recap, DNA damage is occurring constantly in our cells, but our cells have a lot of different ways to repair that damage using different mechanisms for different types of damage that are occurring. DNA mismatch repair takes care of any errors that have been left behind after replication has occurred. Double-strand repair require very different strategy with either homologous or non-homologous recombination happening and joining happening. Homologous recombination is how you would want to repair DNA replication, double-strand breaks, as they would cause a flawless repair without any loss of nucleotides or resulting mutations. And then P53 is the most common mutation that is seen in human cancers, and it is an important integral part of DNA replication and repair control to preserve our DNA sequences. So what happens if DNA cannot be fixed? Well, the cell in that case is either going to stall or it's going to be targeted for death of some sort. There are 3 different types of deaths that a cell may see when there are damages to the cell beyond repair. One, and the most common one that we see, is obviously programmed cell death or apoptosis. Now, in other cases, you may instead see what we call necrosis, which is where a cell gets, just so damaged, it gets an influx of fluid taken into it, which you can see in the form of these multiple vacuoles forming. The cell kind of enlarges until it bursts. The membranes just leak all its content out. However, that's a very damaging death. It damages all the tissue and the cells around it as well. And finally, in some cases, the damage is such that the cell takes it as a starvation response and starts to ingest itself. And it can recover from that state for a very long time. It takes a long time to get to the point of no return. But eventually, in that case, the cell would have taken up so much of itself that there is no way to return. And that process of self-ingestion to death is what we call autophagy. It's also kind of a survival mechanism, actually. So in pathways of necrosis, the main cause of this necrosis happening is because of some sort of stress occurring to the cell causing major damage. And in that case, there are proteins called RIP1 and RIP3 that are activated, or PARP, another protein PARP that may be activated that will then in turn also activate RIP1 and 3. These proteins activate multiple pathways that lead to that whole necrosis response. One of those responses is the release of cathepsin C from lysosomes that release then other kind of lytic enzymes from there as well. Another one is that the mitochondria gets stressed and starts to have an overproduction of reactive oxygen species and depletes the ATP supply in the cell. And finally, you have the endoplasmic reticulum activating calpain production as well. And all those combined lead to that necrosis effect at the end of the day. The second, and the one that's favorable to, the one that we want the cells to die with if they are going to die through a chemotherapy or another response, is apoptosis or programmed cell death. So there are two main ways this can happen. One is through an intrinsic pathway where the cell has received some kind of a lethal stimulus and it internally starts this process of apoptosis. And the second one is through an extrinsic pathway where there are these cell surface receptors or receptors in the membrane called death receptors. Two examples of those are trail receptor and FAS that bind to an extrinsic molecule, maybe a chemotherapy molecule, maybe another molecule that triggers a cascade of reactions leading to the apoptosis. Now, in both cases, one of the last steps that you will see is caspase-3 activation that finally leads to the death of the cell. But in between, depending on which pathway is activated, you will have very, very different steps. In an extrinsic pathway, once the molecule binds to the death receptor and activates it, the internal part of the death receptor will usually activate FAD and pro-caspase 8 and 10. Caspase 8 and 10 then bind to other and activate other proteins, including BID and caspase 3 and 7. BID would then activate further proteins that cause release of cytochrome C from mitochondria that also lead to release of caspase 3 and activation of death. In an intrinsic response, on the other hand, it goes directly into the mitochondrial response. So it starts off by activating proteins like BAX and BAC that cause cytochrome C release. That cytochrome C release is then part of the structure that is made in combination with cytochrome C and caspase 9 to create what we call an apoptosome that activates caspase 9, and then caspase 9 then activates caspase 3 and leads to apoptosis. So whether you activate caspase 8 through an extrinsic pathway or caspase 9 through an intrinsic pathway, the last caspase to be activated is the same. It's caspase 3. And they both lead to the same apoptotic response at the end of the day. So that's where we'll stop. And then next time in class, we are going to start with cell cycle, which is, I don't know what chapter, I think chapter 18. So that's what we will be focusing on next week on Tuesday. Okay?